So let's continue on. What do we have? We have the cerebellum. What is the function of the cerebellum? Balance, coordination, and unconscious proprioception. What were the two proprioceptors associated with this? We said there were two. There was one in the muscle and the second one was in the tendon. The one in the muscle is called muscle spindle fibers. That's for detection of stretch in the muscle. What is the function of Golgi tendon organs? They're found in the tendon and they detect tension in the tendon. Same thing, tension and stretch, but in order for it to be simplified for exam purposes, we're going to memorize it as stretch in a muscle and tension in a tendon. So what are the parts of the cerebellum? Um, how do we remember this? So there is a tree, and we shall call this tree the tree of life. So the tree of life has white fibers passing through them. So they're myelinated. They're likely to be axons, right? Because myelinated axons would send information. But if I have little leaves at the end, we call them foliage, right? So the leaves at the end are a little bit gray on the gray side. So we call them folia. So inside of the cerebellum, we have two pathways. One that transmits information, right? That's called the tree of life, otherwise known as arbor vitae. Arbor vitae is the tree of life. It's for sending information. It has white fibers, so it's for myelinated axons or tracts. And then we have folia. Folia is gray matter, so it has cell bodies for processing information. And imagine there's a little worm, right? And his name is vermis. The worm is crawling up the tree. The worm. So it's called the vermis. So let's take a look at some of the parts that I want you guys to remember. So folia, surface of the cerebellum, high fold neural cortex. It's basically the gray matter of the cerebellum that contains cell bodies for processing information. The worm is in the middle. I will show you what that looks like soon. And then arbor vitae, tree of life, are basically the branches of white matter that pass through the cerebellum. These are the three highlighted points that I want you guys to understand. So I have the vermis. The worm is in the middle, right? It divides the right and left hemispheres. And again, the worm on the hamburger, right? This almost looks like hamburger. I'm sorry, I can't help it. The vermis is in the middle. The folia are the foliage or leaves on the outside. And then let's take a look at it from the inside, and then we'll go back to those questions. So you see these folds. These folds are folia, almost like having let's get back to this, almost like having this, right? The folia or the leaves on the outside, the foliage. So these are the folia, and then the inside is the arbor vitae, the white lines or the tree of life. It almost looks like a tree with leaves. So let's go back one question. What is the little brain? Cerebellum, what are the major parts? So the major parts that I'm going to ask you on are the folia, which is gray matter, cell bodies, the tree of life, arbor vitae, or the white matter for axons and sending information. And we went over this and this. And then there was something that was connected in between the two. The vermis is a landmark structure on the back. What is it associated with? So we said a little brain is associated with balance, coordination, and unconscious proprioception. Nice job. You don't have to worry too much about going over that. Midbrain, we said cranial nerves 3 and 4 and corpora quadrigemina were the four bodies on the backside. And the ones on top were visual, and the ones on bottom were auditory. So cerebral peduncles, you guys can take a look at all the things that I'm trying to highlight. High yield. So diencephalon. Remember we said that diencephalon surprisingly has 3 parts, not 2. So the 3 parts were thalamus, hypothalamus, and epithalamus. Epithalamus was connected to the pineal gland, released melatonin. Melatonin originates from what precursor? From tryptophan. Where do you find high amounts of tryptophan? Inside of turkey. Turkey is a good example. We eat a lot of turkey, so we have high levels of tryptophan during Thanksgiving, and then we have Kanak attack and we want to pass out and fall asleep after we eat too much. Right? Okay, so we went over all of that. We said thalamus. We want you to just remember that it's a relay station. There we go. There are different groups. So if you take the upper division course, you're going to be required to understand this. But for the purpose of class, we're keeping it strictly to relay station. But look at all these groups. There's 5 different groups, and they go from different parts of the brain to communicate sensory information to and from the thalamus to the brain to different parts. So don't worry too much about it for my class. And here we go. This is a review of the information that you've gotten up until this point. We did this. Oh, that's really ugly. Okay, let's try that again. Let's try that. Oh, that's still ugly, but that's okay. So we had fore, mid, hind, pros, mes, rom. Pros became tel, die, mes, met, miles, right? Tel became the 5 lobes of the big brain or the cerebrum. Die became 3 things actually, so we're going to write all 3. It became the thalamus, the hypothalamus, and the epithalamus. And that's not, oh, that's good. See the pattern? I don't know. And then mes stayed as midbrain. That was cranial nerves 3 and 4. This one for the big brain was frontal lobe, temporal lobe, occipital lobe, the parietal lobe, and the insula. And then we have lateral ventricles. So we're going to make a reference. Lateral ventricles are about the level of the cerebrum. And then diencephalon is third ventricle. And then pons and medulla, pons and cerebellum are about the fourth ventricle. The cerebral aqueduct actually passes through the midbrain. And if you guys are like, what's going on? Go back and look at the chicken. There's actually even a diagram that we kind of went over. And the diagram on the PowerPoint slide just classifies all of it. So the thalamus is relay station, right? The hypothalamus is sleep, sex, hunger, thirst, temperature regulation. And then epithalamus was melatonin, not melanin. Be careful, melanin is skin pigment. And then pons was 5, 6, 7, 8. Who do we appreciate? Pons, yay, pons. And then cerebellum was for balance coordination and unconscious proprioception. And then we have at the very bottom, the medulla was 9, 10, 11, 12, and swallow, cough, vomit, cardiovascular centers, and respiratory centers. Whew, it's a lot of information, but you guys are doing really good. So same thing. Parts of the primary vesicle, that's what we looked at here, right? And then the next one was, what are other names used? So that was essentially the forebrain, midbrain, hindbrain, and what are they also called? What are the parts of the secondary? So this was primary, this is secondary, and this is the adult structures, right? That's the final adult structures that they become. So forebrain, midbrain, hind, you guys can review that. I just went over it right now. The fates of the secondary brain vesicles. Telencephalon becomes the big brain. Diencephalon becomes the relay station and the hypothalamus and the pineal gland, right? Mes becomes midbrain is cranial nerves 3 and 4. Metencephalon branches into 2 things, the pons and the cerebellum. Cerebellum for balance coordination and unconscious proprioception. Pons is for 5, 6, 7, 8. Who do we appreciate? And then myelencephalon is the medulla oblongata for cranial nerves 9, 10, 11, 12. You guys can go back to what the other things were. What are the major lobes of the brain? We did that. We said there were these 5 that we went over. Frontal lobe for social norms, temporal lobe for audition, right, for auditory, occipital lobe for visual cortex, parietal lobe for sensory or sensations, and then insula is the landmark structure for the basal nuclei, which initiate movement. The sulcus, central sulcus. If I didn't do this already, I don't mind repeating. It's nice to repeat the information because it's highly repetitive, and the more you can repeat, the better off you are. So we have the brain. It looks like a baseball mitt, and then we have the meaty underside, and we have the brain stem. Okay, so this is the frontal lobe. There's a little dip that connects all the way down. We call this the central sulcus. And then there's elevation in front and elevation in back. The one in front is called the precentral gyrus. The one behind is just called the postcentral gyrus because it's behind the central sulcus. So it's a landmark structure that kind of separates the 2 different functions and the 2 different lobes of the brain. The one in front, frontal, is also associated with MEV. So I probably should have drawn this a different way. No, this is right, SAD-MEV. So we have our mnemonic over and over again. I told you that your final exam is going to have SAD-MEV over and over and over again. So SAD stands for sensory afferent, is located dorsal. Doesn't posterior Isn't that another word to describe behind, back, or dorsal? So if this is in the back and it's sensory, this is your primary sensory cortex. And then if I look at the front, MEV, motor, efferent, ventral. So ventral also means pre, so it can also mean anterior or in front. So in this case, the frontal lobe is associated with your primary motor cortex. So these are the questions that we're answering right now. What gyrus is found in the front? So it's MEV, it's motor. What gyrus is found behind the central sulcus? It's SAD, so it's sensory. And then what are other functional areas of the cerebral cortex? What general functions are associated with motor areas, sensory areas? We kind of already did that, right? We did primary motor and primary sensory. And then what is Alzheimer's disease? So Alzheimer's disease is a progressive degenerative disease that leads to loss of memory. So loss of short-term memory. And we're going to simplify it down to that. Some of the key features that you actually cannot diagnose Alzheimer's until post-mortem. So to confirm that the patient has Alzheimer's, they have to see something called amyloid plaques. And amyloid plaques are little protein bodies that are found inside of the brain. And if you see these amyloid plaques, then you can definitively diagnose Alzheimer's. Up until that point, it's projected that they have Alzheimer's, but they don't quite have a definitive diagnosis, a way to say clearly it is. But there are signs and symptoms that are related to it.